when it eventually gets going. Right, that's on record. Right. Okay, no, there we go. Hello, everyone. Welcome to today's 5th session on BGA24. Today, we're going to have a session on BRAKER3 by Katharina Hoff. So please take it away.
Thank you, Damon. Good morning, everyone, from my side as well. I hope you have seen in the chat that we have some essential resources. In particular, we have a Discord channel. If you want to drop questions, you can put them in the Discord channel. Of course, you could also unmute yourself here if you want to ask something. I'm not someone who feels disturbed by someone unmuting themselves in the middle of my talk. That will be fine. Okay, I try to go to presentation mode. I assume all of you can see my screen sharing. I hope so. So my plan today with you, thank you for joining, is that first I will give a little talk where I explain to you, I try to get this, oh, the recording bar can go away, okay, where I explain to you how our gene prediction tools generally work, like what are the expected inputs, why do we do it this way? And then we really want to dive into practice. For diving into practice, I think you've all been informed that you need a GitHub account in order to start our session on Gitpod later. Don't worry, I'll walk you through that, but the account you need beforehand. Okay, so the topic is mainly structural genome annotation with BRAKER3, but we will also dive into a couple of accompanying tools today. And I'm trying to switch slide. Okay, good. This is my very professional Wikipedia style figure on a structural genome annotation problem. So let's look at the top panel. Yeah, your input is eukaryotic DNA, genomic context, genomic scaffolds, chromosomes. And in there somewhere, but you do not know where yet, you have protein coding genes encoded. And they consist of coding sequences, those are the red things, that are sometimes, but not always, interrupted by so-called introns, that's the gray things. This all is surrounded by a lot of other stuff, like there are untranslated regions shown in blue here, there are promoters, enhancers, silencers, and we do take these surroundings into account with our methods, but the whole problem is for us to find the red parts, the coding sequences that belong to a particular transcript. And it could also be several transcripts in one place that belong to the same gene. So this is typically in the cell transcribed into mRNA, yeah, but in the mRNA, you still do not know what is UTR, untranslated part, and what is coding sequence. The introns you can usually figure out on the mRNA level because they get spliced out by the cellular machinery. So if you would sequence this thing here, most of the time you catch it without the introns already. You have the mature DNA. And this is ultimately then by the cell translated into a protein. And this is why we do what we do. We want to find the protein coding genes on the genome level in order to predict what the proteins look like, because that's what we all want to know to understand biology. So for us, if we look into how to annotate a genome, we have the information for genome annotation from the genome sequence itself. With the genome sequence itself, we can try to train all kinds of different models. In this case today, we're talking about hidden Markov model style models. They are mathematical models. It could also be other models. We can train them to recognize the coding sequences. What we can also use in our pipelines is the information from this step, like from the lowest red arrow style here. That's the mature mRNA. That is what you commonly observe if you do RNA-seq. This is mostly done today when you want to annotate a sequence. So we can project that back to the genome. And then also we have, over the past 2 decades, accumulated a lot of information on protein sequences from eukaryotic genomes. And we can also use that. So this is also an information because we can also project this thing here, even though it looks like a weird fuzzy ball, back to genome level. I said mathematical models and signals and so on. So what is important to us? Again, we're here at the top looking at the genome sequence. And if the cell wants to transcribe an mRNA, it will know where to do that. There is some kind of signal for a transcription start site where the RNA polymerase will get started. It will also know where to fall off from the transcription, right? So there must be a transcription termination site. These patterns, they are in some cases even visible to the human eye if you know what you're looking for, but they can vary quite a lot across different species. So these signals for transcription, they're pretty species specific. I mean, if you have two close relatives, maybe it looks very similar, but if you look at the human and let's say a fungus, it can be very different. Then what happens in the next step is, as I said, the translation for protein. So there's again a signal. There's a translation start site. There is a translation termination site. It's typically called the stop codon. And again, the cell knows how to recognize that with the ribosome to get started and to stop. And we can also use that in mathematical models to predict genes. The introns, the things with the little arrowheads here in between, they also have typical signals. There are highly conserved donor and acceptor splice sites. And if I say highly conserved, I do not necessarily mean that they are all the same everywhere in life, but they are pretty conserved. And also there are things like the branch point region. So these are signals that we can use to inform some kind of model, like how does a eukaryotic gene structure look like on genome level. But there are also other signals that are not typically like patterns. For example, the coding sequence region is under a different evolutionary pressure compared to, let's say, non-coding regions, like flanking regions of genes. If something mutates in the flanking region, the effect on protein sequences is not as drastic as if you mutate in the coding sequence, right? So therefore, we can also use something like polynucleotide frequency patterns in order to distinguish probably coding region from probably non-coding region, and that is also used. As I said before, these signals altogether, they are pretty species specific. They are not completely universal. And in the tools we talk about today, that means that we have to adapt the model to the species. And this is what it's all about. Yeah, we have to find training genes. Like before we know the genes in the species we want to annotate, we have to find training genes, then we train the model, and then it's hopefully going to work well. So to make it a bit more clear, what is the structural genome annotation problem? We have as input a genome assembly and probably some kind of extrinsic evidence. I would say as a minimum level today, we have a protein sequence database. Even if it's poor, like for protists, I know it's not great, but we have something. And in many, many cases, we have mRNA-seq data as well. And what we want to do with these two inputs is to produce an output, which for me is like a table. We have some formats like GFF3 and GTF. And these tables, they describe where, on which sequence do we have our coding sequences and our introns. And you can use these tables in order to extract the transcript sequences, the coding sequences, and then translate that into protein sequences. This is also what's shown at the bottom. Basically, there will be a ground truth, but you usually do not know the ground truth. That is the black thing here, right? In some species, a lot of effort has gone into annotation, but if you annotate a novel genome, you don't have the ground truth. And what we want to do is to come as close as possible to the ground truth of what is a correct gene structure. What we will do is we will use the protein to genome alignments. They are spliced alignments. And we hopefully always have many informant proteins, so we have a lot of data here. And also RNA-seq alignments, usually also a lot more information than shown here. This is a very simple picture. And then we put that into our gene prediction tool to infer the hopefully correct gene structure, but of course it will carry errors, as you can see if you compare the red and the black track here. I'm just trying to have an eye on that Discord channel. Nothing showed up yet. Okay, so our problem is basically that if we want to annotate a novel species, we have not seen the gene structures before. It's a chicken-egg problem. By the way, that chicken there, that is Bianca. She was my mom's chicken. She passed away last year, and that is actually her real egg. It looks super white, but I swear to God, I took that photo. Okay, so in order to solve that little chicken egg problem, we bring our gene finders to the table. Yeah, we have BRAKER and GALBA developed, and they are fully automated pipelines that solve this problem of generating the training examples, then also annotating the genomes. Zebra is a little bit of a different story. Zebra is a tool that helps you to combine different gene sets with evidence. And today you see my face here on the camera, but I'm just the face presenting this. BRAKER in particular is the work of two international teams. On the Greifswald side in Germany, where I'm sitting today, Lars Gabriel was the main developer of BRAKER3. And Mario Stanke was the one who actually obtained the funding for this together with Mark Bordowski from Georgia Tech. They are the team that develops GeneMark tools, and Alex Lomsatze. And then there's also Tomasz Brunner. Tomasz was a PhD student in Georgia Tech. He's now at the JGI, and he's still very active in the methods development. Now, everything started for us with BRAKER1. And this was the first pipeline that we were aware of that was designed to solve this training gene problem using RNA-seq short read data. And back then, we didn't have the correct mapping tools or the good mapping tools that we have today. We had like other mapping tools. We didn't have so good assemblers as today, and we didn't have so good data as today. So what we had figured out back in 2014-15, before we published this, is that the information from the raw spliced alignments, like from the mappings, was the most valuable in order to inform a tool called GeneMark ET to build the training genes for our pipeline. So that was the special thing about this one. I'm telling you about it because in some cases you still want to use BRAKER1 today, okay? Even though we have BRAKER3. So then we use the GeneMark ET predicted genes in order to train the Augustus gene finder. That is a gene finder developed by Mario Stanke. And then we use Augustus to predict genes with the evidence from the RNA-seq data. And originally that was it, but in recent years we have started to combine the GeneMark, the good genes that we use for training Augustus, with the Augustus genes with Zebra, the transcript selector for BRAKER, and then you get a final gene set. The special thing, as I said before, is that you don't need a transcriptome assembly. So this is still a to-go-to pipeline if you don't have very deep RNA-seq data, or if BRAKER3 fails you for some other reason and your genome is not very large. I want to quickly explain what happens in there again. So this is about the step that GeneMark ET is performing. So GeneMark ET is a very unique kind of tool. It's a hidden Markov model-based gene finder that performs self-training. We were never able to pull that off for Augustus. And they use the RNA-seq spliced alignments that you see here. They are those weird triangular-like things here. Those are spliced junctions. They use this to feed back to GeneMark during the model refinement. So it's not used for prediction. It's used to make the model better. And this is why it was like a really good combination with Augustus, because Augustus could not do the self-training. It didn't care about the evidence during the training at all. It does it in a different step. So GeneMark ET employs the unsupervised training. It includes the training introns from the RNA-seq into its training step, but it does not use them during prediction, and it does not require that the RNA-seq data is deep enough for assembly. What Augustus, on the other hand, then downstream does, we use the same evidence, but we use it for the prediction step. And Augustus can, in contrast to the GeneMark tools, predict alternative splicing isoforms. You see an example here where there is a possibility to have a longer and a shorter coding sequence, and that is supported by these spliced junctions that come from real RNA-seq data. Augustus needs prior data for training, so we can build on the genes that GeneMark can predict, and it uses the RNA-seq information for the prediction step. Now, if we talk accuracy, we first have to talk about limitations, maybe. BRAKER1 is a tool that works well in small genomes, and let's call them medium-sized genomes. If you have like a mammalian genome or like something bigger than 1 gigabase pair, it's probably not going to work very well. Please keep that in mind. Yeah, this is for small and medium-sized genomes, but in there we are, if we look at average gene prediction accuracy on gene level, we are at around 40%. Yeah, ballpark numbers. Spoiler, if you can, you should probably use BRAKER3. Reasons to resort back to BRAKER1 is, as I said, if you cannot assemble transcripts. Yeah, if you don't have deep sequencing data, then BRAKER1 may be an option if your genome is small or medium-sized. Now, let me take it to the next level. We developed BRAKER2. And the ideas were initially very, very similar. But instead of using RNA-seq data, we use proteins. And I don't mean proteins of the closest relative. I mean a huge database. We're usually using OrthoDB today. but we also played with eggnog. You can basically use any huge database. What's important is that you need proteins from more than one informant species, because we look at how many proteins map to a locus, and if you input only one relative, that would not work, right? The tool from GeneMark's side here is called GeneMark EP+. It works quite similarly, has advanced a little over the GeneMark ET. And otherwise, the pipeline is very, very similar. It is today, for many people, to go to tool if you don't have any RNA-seq data. Yeah, then you can use BRAKER3. I said GeneMark EP advanced a little. It really did. A few special features that came into the GeneMark pipeline with this is that Georgia Tech developed a way to score start and stop codons derived from the protein to genome spliced alignments. And then they adapted GeneMark also to correct, if necessary, the start codon position or also the intron positions according to the evidence. So GeneMark EP+ does not only use the data during training, it also uses it during prediction. Augustus pretty much works the same way as in BRAKER1. Again, ballpark numbers is if you have a small and medium-sized genome, you're probably on average on gene level at like 40% accuracy. Okay, then we tried for a long time to figure out how to use the large protein database and the transcriptome data simultaneously. This was not an easy nut to crack. On the way there, this is not like the ultimate achievement, but on the way there, we developed the Zebra. It's called the transcript selector for BRAKER. And the idea here is basically that you run BRAKER1 with the RNA-seq data, you run BRAKER2 with the protein database, and you combine those gene sets in a good way according to the evidence so that you get the best gene models of all of this data. Zebra cannot only run with BRAKER gene sets, it can also, for example, use GALVA gene sets. If you format any other gene set in the correct way, like if you make it look like our GTF output files, it will also work with others, of course. We showed that Zebra can increase accuracy. So the initial idea was that you use BRAKER1 and BRAKER2 for this, but as I have already shown you on previous slides, we even put it inside our pipelines to increase accuracy. So this approach here, this would still be a to-go-to approach if you cannot use BRAKER3. If you use Zebra in this way for a small or medium-sized genome with BRAKER1 or BRAKER2, you will like step up your accuracy a little bit, let's say by 10%, which is good if you cannot do anything else. But of course, we want to do a lot better. We're not paying attention to the hand here. We're paying attention to the heel. With BRAKER3, we want to get to the 70 to 80% ballpark accuracy. So BRAKER3 is our pipeline that is for genomes of all sizes. It also works for the big ones. And it uses RNA-seq and protein data simultaneously. So this is the outline of our pipeline. You basically now have 3 inputs: the genome sequence, the RNA-seq file, and the protein database. And then the underlying tool in there is GeneMark ETP, which uses both at the same time. A lot changed under the hood. We're now assembling the RNA-seq data, but otherwise the pipeline looks pretty similar to the other ones I showed you. But the main difference to BRAKER1 is that you assemble the RNA-seq data. So I said a lot changed under the hood, and I meant that. Let's for a moment look only here at these 3 inputs: the RNA-seq file, the protein database, and the genome file. So what we do is, with the BEM file, with the spliced alignment file from RNA-seq data, and the genome file, we do a genome-guided transcriptome assembly with string-tie2. Then we take those transcript sequences and we train a self-training gene prediction tool for transcript sequences on it. It's called GeneMark ST, and it will call ORFs in those transcripts. Regrettably, it will not do that with perfect high accuracy. That's why we have kind of a denoising step afterwards. We use the protein database in order to remove noise, fix some gene models in the transcripts. And then we can, since string-tie also gives us genomic coordinates of the transcripts, project the gene models back to genome space, and we can train the eukaryotic GeneMark. And this will produce 3 important output files. One is like the file with all the GeneMark gene models, which are quite good already. Another one is the file with the best ones of them, like the most reliable ones. That's the training GTF file. And then also GeneMark now produces our hints file for Augustus. Augustus is then trained on the training genes, produces an output together with the hints files, predicted genes, and we combine with Zebra. In Zebra, we enforce that those really good training genes are maintained in that gene set. And we also put in some genes from GeneMark and from Augustus according to the evidence, and we get our output. Now, when I talk about accuracy, we have to talk about how can we measure it, right? So what we typically do is we go to some well-annotated genomes where we kind of trust the annotation. We selected for the slide I'm going to show next 3 small ones. It's the tail cress, the nematode, and the fruit fly, and 3 bigger ones, the chicken, the mouse, and the tomato. They vary in size. Yeah, you see the small ones are like 100 MB, and then we have like a 2 GB or more than 2 GB with the mouse. And I show you here on the right how many gene models have been annotated. It's kind of important to keep in mind that these annotations are not perfect. Like they will still carry some errors, but we can at least attempt to measure according to that. And then we measure 2 things. We measure basically the sensitivity. Some people also call it the recall. It's the percentage of correctly found genes, transcripts, exons if we run our tool and we compare to the reference annotation. And then we can also measure the specificity or precision, and that's the percentage of the correctly found features in our predicted set if we compare it to the reference annotation. And we can average that with a harmonic mean, also called F1. Those were the values I showed you on previous slides. Now, here you have a plot for the small genomes. And we measure like 3 different scales. We measure on exon level. Let's look at that first. Everything looks similar on exon level. BRAKER3 is the red square. Yeah, the square thing is BRAKER3, but everything else looks pretty similar. Then we measure also on gene level. What does that mean? So exon level means that we compare individually every predicted CDS exon with every annotated CDS exon when we count what's a true positive and so on. Gene level means that we take everything that is predicted and that matches one possible isoform of a gene correctly in the reference annotation as a true positive. So here it would be enough if we just match one of the isoforms from alternative splicing to count true positive. On transcript level, we basically look at every alternative splicing isoform on its own. So it's harder to get that one right. Now let's focus on the gene level plot. That's possibly the most important one. And what we compare to mainly here is the GeneMark ETP, because the GeneMark ETP is an intermediate step in our pipeline. Yeah, and we see that here we get like an improvement in specificity and sensitivity that is substantial. We also, for sake of completeness, show you BRAKER1, light blue triangle, and BRAKER2, that's the beige triangle or brownish, and the zebra step. Yeah, the zebra step is the orange thing. And remember, I told you before, if BRAKER3 fails you for one or the other reason, then you can resort to doing the BRAKER2 and BRAKER1 with zebra combiner step, and you also get a reasonable accuracy if your genome is rather small. Now, if we look at large genomes, that's a bit of a different story. Because BRAKER1 and BRAKER2 do not work well in large genomes. So let's again focus on the BRAKER3 accuracy. That is like the red rectangle up here. We're on precision or specificity level a bit below 80%. On sensitivity or recall, we are at 70%. And that is again like 8 to 10% improvement over the intermediate step of GeneMark ETP. So the takeaway message here is, if your genome is large, use BRAKER3. If your genome is small, use BRAKER3, but if it doesn't work, you can still do BRAKER1, BRAKER2 with zebra. So this here is like a bit of an outlook to our hands-on session. It's the BRAKER command. So BRAKER has several different entry points for the RNA-seq data. Today, we will actually use BEM files as input because we don't want to like download complete RNA-seq libraries from the sequence read archive. But you could give sequence read archive IDs, and BRAKER would like auto-download, auto-align, and so on. Or you have the BEM files or FASTQ files sitting on your hard drive already, then you can provide basically the name stems of those files and the directory where they are. Most important input is the genome file and, of course, the protein database file in FAST8 format. And then there's something I'm not going to go into detail today that is new. We have an option where you can provide the BUSCO lineage. So what we noticed over the years when we run our tools is that we tend to overlook certain single exon BUSCOs. BUSCOs are supposed to be present in your annotation. You want them to be there when you submit that gene set to a public database, but we're not good at finding them sometimes. So we included running CompLeasm. It's a tool developed by Neng Wang, sorry, I mispronounced that first, and Heng Li, to detect BUSCOs, and we make sure that they end up in our final gene set if you invoke that option. Of course, you should probably not invoke that if your goal is to compare different pipelines, how well they can detect BUSCOs, because we really put them in there. But this increases BUSCO detection a little bit. Now, in terms of runtime, to give you ideas, today we will have something that runs 8 to 10 minutes on the Git pod. But if you have a real genome, on average, across all the genomes that I showed you, if you have a machine with 48 threads with BRAKER3, you would be in the range of 18 hours runtime. Of course, it depends on how much RNA-seq data you put in there. Yeah, and of how much protein data. For example, sometimes people use the complete OrthoDB. That increases runtime a little bit. If you partition it, if you use only the vertebrate OrthoDB for mammalian genomes, it's a little faster. If you have like 50 RNA-seq libraries, you will probably have a lot longer runtime because they all need to be downloaded and aligned. If you use only 6, it's going to be shorter. But this is ballpark numbers. Of these 18 hours, about 6 hours, like 1/3, will be consumed by the GeneMark ETP step. And if you think about going back to BRAKER1 or BRAKER2 on these data is like in the 9 hours ballpark time, and BRAKER1 in the 6 to 7 hours. You can get BRAKER3 from GitHub. However, I would nowadays strongly recommend that you get it from Docker Hub, because we have like put together a container that we're also using today where we make sure that all the different software versions are actually compatible with each other. I have to say, I don't recommend you go the Conda way, because in Conda, we don't maintain the BRAKER recipe ourselves, and there are always inconsistencies and problems showing up with different versions of tools. License-like, if you are in academia, BRAKER3 is free to use. If you are commercial, you have to keep in mind that GeneMark ETP is under the attribution non-commercial share-alike 4.0 international license, which basically means please talk to Mark Wardowski and Alex Lomsatze if you want to buy a license. Okay, so I'm going to move on to the Galba pipeline. Galba, by the way, is completely free. The team changed a little bit here. Galba actually was kind of triggered by Heng Li, the developer of MiniProt. It's a protein splice alignment tool. It's very fast. And then we quickly got together with Tomas Brunner, who's now at the JGI, and Jolif Garland from New Zealand, and Lars, and from our group, Natalia, Mattis, and Mario. And we made a pipeline that was simply trying to use MiniProt in order to make really good gene sets with Augustus. And then at some point in time, Ethan Tolman, Paul Franzen, who never saw me in person, had a really brilliant idea how to filter those gene sets. So that's how they came into the picture. And Daniel and Steffen, they are basically developers of some of the Python components that we use in there. So MiniProt is, as I said, a protein to genome splice aligner, in a way comparable to Spawn, if you have heard about that. And the result of MiniProt is basically already a file with gene models, but MiniProt can only detect so much. It will overlook some things because maybe they were not in the donor database, maybe the alignment step didn't work for some genes. So that's why on top of this, if you want to annotate a genome, it makes sense to use a gene finder such as Augustus. And when we started out with this, we thought it should be really easy. We can just train Augustus on MiniProt and then we'll have a great gene set. That was the idea. One of the big questions at the beginning was, do we really need another pipeline? Don't we have enough with BRAKER? And by the time First of all, we didn't have BRAKER3, so we needed something for the large genomes. And also there are a number of cases where you fail to obtain RNA-seq data for various reasons. So yeah, it was enough reason to make it. Also, there's always a demand from some users that you want to annotate a target genome only with the proteins of the closest or 2 or 3 closest relatives. Yeah, there are situations where this makes sense, and Breaker is not particularly good at that. And as I said, I really wanted to have a fully open source tool that's free for everyone. So it turned out to be a bit more complicated than anticipated because miniprot is a little noisy. So we had to develop our own filters called the miniprot hint. And the miniprot hint increased accuracy quite a lot. And then we also had to change our selection of training genes. Using every one was like from the gene candidates was not a good idea. So we decided to use the best one per locus, discarding alternative isoforms. We should have considered that from the start. That also increased accuracy. And then we also thought of how does GeneMark work? I mean, it does this self-model refinement, and we kind of implemented something like that. We call it the iterative training, which means we have Augustus predict a gene set, we filter it, then we train again, we predict again. It costs a bit of runtime, but it increases accuracy, of course. And then Ethan's contribution was basically that when we output our gene set, it's noisy. And we have as input proteins from close relatives, so why not use them to denoise the data set? That's the diamond filter down here. So we did all the development in small model species. That's the fruit fly. Galba is not a pipeline for the fruit fly. This had just like runtime reasons. And what I show you here is that we use the red thing, a very, very, very close relative as protein donor up here. And then the combo thing are 5 different Drosophila species that are not as similar. This is maybe a more typical case if you annotate a novel genome. Yeah, you combine a couple of close but not that close relatives. And as you can see, all these development steps actually tended to help in both cases. This super confusing but beautiful plot shows you how Galba, BRAKER3, BRAKER2, and also kind of a competing pipeline, the FunAnnotate, perform in a number of genomes. We have here different insects and a spider. Then we have here C. elegans standing kind of alone in the metazoa. Then we have a couple of plants. Here, these are all plants. And then we have a couple of vertebrates. So BRAKER3 overall has the highest accuracy. So if you have RNA-seq data, please use BRAKER3 if you can. But if you have no RNA-seq data, there are scenarios where Galba is clearly superior to, let's say, BRAKER2, because BRAKER2 simply does not perform well in the large genomes, as I said before. And you can also see that in all cases, we perform better than FunAnnotate. So with Galba, ballpark numbers in terms of accuracy, we are somewhere around the 40–45%, even in large genomes. So if you have no RNA-seq data, but you have proteins of a close relatives, this can be a good pipeline for you. Coming to how do you know? Like once you've run it, how do you know whether you did a good job? Maybe you should use another pipeline, you know? There are different things. The first thing is that I always recommend you actually set up a genome browser instance. You can use any genome browser. Use JBrowse too. I think there is a session here in the BGA about that. But there are many genome browsers. I prefer the UCSC genome browser with my makeup script, but that's mainly because I made the makeup and we have a connection to UCSC from Mario. You can use any genome browser. What you want to look at is your gene models and all the evidence that went in. Yeah, and you want to jump to random loci and you want to look whether these gene models make sense in context with your evidence. Sometimes you can quickly observe if you jump to, let's say, 20 loci that you have, for example, a split gene problem or a tandem joining gene problem. And then you don't want to like keep this gene set as a final gene set. The other thing is that you can describe your annotation. Now with simple statistics, like you can count the number of genes, count the number of transcripts. You can count the ratio for monoexonic to multi-exonic genes. For plants, supposed to be 0.2, I think. I'm not 100% convinced it must always be like that, but Jill Begerson's team said for plants it should be like that. What you can do if you don't know about your target organism, you download all the reference annotations of the relatives and you compute the numbers for them too, so that you have something to compare to. Also, maybe check who made those annotations. There's a big variance according to methods. You can compute median number of exons per transcript, maximum number, median transcript length, and so on. So compute these numbers, and if possible, compare them to the close relatives so that you know whether you ended up in the right ballpark numbers. And then we have two pipelines that help you assess sensitivity according to clade-specific conserved marker genes. The first one, that's the older one, is BUSCO. BUSCO will show you how many of your clade-specific marker genes you have complete. It will differentiate in complete and duplicated, single copy and duplicated. Be careful with that, because BUSCO does not handle alternative splicing isoforms. Yeah, it will count everything you put in as a separate thing. So when we predict alternative splicing, we usually get quite a high duplication number, and it doesn't mean that we have duplications of the marker genes. It's just alternative splicing. It will also show you fragmentation and missing. And you can compare that across the different levels, for example, if you run BRAKER1 and BRAKER2 of all the tools in the pipeline, but you can also compare this to like the other species in your clade that have been annotated to see whether you're ending up in the right range. BUSCO has very different numbers of marker genes depending on your clade. I think it has like thousands of them for plants, for example, or insects. That's great. But if you move to protists, for example, you're there with 100 marker genes, and then it gets a little questionable how much value does it have. There's a new kit on the block that's called OMARK. It also measures basically the presence of marker genes. They are here called not BUSCOs, but HAWKs. You see like the plot from their website here. That's not my plot. And they will also give you like the green thing is like what have you found? The red thing is what you missed. And OMARK, in contrast to BUSCO, can handle alternative splicing isoforms, which is a nice feature. Yeah, it will show you true duplications if you do the input right. It can do a lot of other things too. It can tell you how many of your proteins were placed consistently according to the lineage, like whether it matches with the database knowledge and so on. And very interesting for me, it shows you contamination. When I work on straminopiles, that is always very nice to see whether something has been contaminated. Okay. Now, I would like you to move on to the hands-on lab session. That means you have to get active now. You have to start your Gitpod. I recommend before you start your Gitpod sessions that you go to GitHub and you log in so that GitHub knows who you are, right? And then you can either click the link on the slide, or maybe we can put that in the chat. Maybe Damon can put it in the chat. I don't know, can you? So that you have less trouble finding it. And I will now wait a little bit, and I will go to the Discord channel while you all move on to the Gitpod, okay? And I will answer questions. So what do I see here? There was a question. It's called, is there a minimum number of gene models that Augustus requires for training and prediction? How does it determine what constitutes high-quality gene models as the training GTF file? So the first question, minimum number of gene models. You can, in principle, train a tool like Augustus on 60 gene models. The problem is that if you do that, these 60 gene models are not going to represent the whole diversity of how genes look like in your species. So the resulting model is just going to be a bit crappy. We have, for the BRAKER2 publication, in the supplementary material shown for Arabidopsis, and I think also for other species, how the ab initio prediction accuracy of our tools increases if you increase the number of training genes. So there is the supplementary figure on that. And if I recall correctly, it's a good ballpark number to have like 5,000, 6,000 training genes. This actually came a bit surprising to us because we always thought fewer would be sufficient. So having a couple of thousands would be good. A true minimum is hard to define because, I mean, you can do a lot of things. The question is whether it makes sense. Now, the second question was, what is a high quality gene model? This depends on the pipeline. For BRAKER1, we basically took everything as a high quality gene model that has an intron RNA-seq coverage of 10. Yeah, if there were 10 spliced alignments to every intron in a multi-exon gene, we would say that's a high quality gene. We were not able to determine which single exon genes are high quality, so we did random sampling of the single exon genes back then. For BRAKER2, I think we said we have to have a minimum of 4 in the splice site junction coverage, 4 protein to genome splice alignments in all introns. And then Tomasz Brunner developed a scoring system to score the spliced alignments. It's called PROTINT. We also implemented in the mini PROTINT the same scoring approach. Basically, he uses this coverage information in combination with now this called the IBA, intron border alignment score. You can check these methods in the GeneMark EP paper. It's all described in there. So we use these scores in order to determine whether a gene is a high quality gene. In BRAKER3, that's pretty similar. But here we, in BRAKER3, it's actually a little different because in BRAKER3, we go the string tie way, right? But in the end, it kind of burns down to a similar selection process. Then there was the question, there's what genome size is typically considered large? Large is for me more than 1 gigabase pair. Yeah, that's large. Then there was a different question. Do you see a correlation between position on the genome and quality of the predicted gene model? For example, closer to the centromere, less accurate genes. I honestly have never analyzed that systematically, so I sadly have no answer for you on that one. I would expect that the centromeres are masked by the repeat masking. We will come to that in the hands-on session. So I don't think the centromeres play a role because they will be masked out, but I have never systematically looked at it. Okay, the genome size question we answered. Then there was another one. GALBA generates a lot of gene models, sometimes too much compared to BRAKER genome threader integration. Can you comment on that? This should not be the case anymore if you use GALBA with a diamond filter, Sam. I don't know whether you did that or you did not, but they should be removed. So the genome threader thing, it like fell down from the sky on some people now. So a long time ago, we had a pipeline that is similar to the GALBA pipeline, but it doesn't use miniprot, it used genome threader. Genome threader is also a really amazing protein to genome splice alignment tool, but it has a much lower sensitivity. This is maybe also one of the reasons. Was there a question from Hunger Coffee Anderson? No? Okay. There's this intrinsic answer that genome threader has a lower sensitivity, but there's also the compensation method. And then Mike says, can BRAKER3 work with only protein evidence? No, BRAKER3 needs RNA-seq evidence. If you call BRAKER with only protein evidence, you call BRAKER2 basically. If so, has it been compared with GALBA? I did show you that. I showed you a plot. You can go back to the slides or to the publications for that. Yes, it has been compared. The summary is basically that in the large genomes, GALBA works better than BRAKER2. Then there was another one. If you have no RNA-seq for prediction, would you suggest use BRAKER2 or 3 or GALBA? So if you have a small and medium-sized genome, let's say smaller than 1 gigabase pair, I would run BRAKER2. If you have a large genome, I would run GALBA. The thing is, we always do our benchmarking and assessment with the OrthoDB protein partitions. You can simply concatenate proteins of your close relatives to the OrthoDB partitions for BRAKER2 to also boost accuracy a little bit. Just concatenate the files. Okay, so naive question. How does BRAKER3 compare to the GNOMON pipeline? Oh, wouldn't I wish to know? I really would like to know. The thing is that I cannot run the GNOMON pipeline. I tried to install it, believe me. I spent like 2 weeks on it. Is it worth it to annotate a genome already processed as RefSeq? This is a very interesting question. And I would say it totally depends. We have very recently, and it's not published yet, annotated 49 diatom genomes. These genomes had not been previously annotated. And then we compared our own annotations to the existing RefSeq annotations of diatoms. And then we saw some really funny things in those annotations. So I would say this is a very difficult question to answer. Sometimes the RefSeq annotations are great, but they are not all great, and maybe it depends on the clade you look at. Okay, then there's another one. Should you mask your genome, soft or hard, before putting it into the software? Soft, but we will come to that. Now, I hope I answered all the questions. Does someone want to unmute themselves to ask another one?
I think you have a few more being typed in.
Aha, then we stay on that. It's okay.
I have a question, Katarina. How does BRAKER3 work with UTRs? Like, how does it compare against? Because I'm guessing with Galba, you don't get any UTRs.
Yeah, the UTRs are a neglected child of ours. We don't have a good working pipeline for the UTRs. That's like the short summary. We have some buggy script, but it's not doing the proper job. And basically, we have nobody working on that at the moment. I have an undergraduate student who's like trying to fix it, but I'm not too optimistic we will have a solution for UTRs that works well anytime soon. So we generally predict without UTRs.
Brilliant, thank you.
Okay, there was another one. No, I don't see another question. That was the masking question. Okay, good. Now, I hope that all of you have landed on the website where I am right now. That is like the startup website for the Git pod. And what I want you to pay attention to is that you can boot two different kind of instances. You want to boot the large one, okay? Boot the 8 cores, because otherwise your BRAKER3 job is gonna like run longer than for everyone else, yeah? So if you're on this page, select large. And I think you should automatically have the VS Code selected. That should be fine. And then you press continue. And maybe I should have pressed continue before I started answering the questions. If you do it for the first time, to start pulling the container for like a minute. But I did start it yesterday, so my container should be there pretty soon. And if yours has succeeded booting, let me know in the Discord channel, okay? So that we get an impression.
You actually have a couple of questions on the YouTube stream.
Okay, I don't see them. Can you read one?
Yeah, sure. So this is from Mohammed Salman. Question 1. I have Illumina paired end RNA-seq data from 3 tissues, the head, thorax, and legs of the same species. Should I use one RNA-seq sample or merge aligned sorted BAM files with HiSat or BRAKER3? Quite a question.
So you want to use all of these tissue data, definitely. So if you have like one file for each of these tissues, use them all. If you did like 20 replicates for each tissue, you can randomly select some of them. You don't need 20 replicates per tissue, okay? I don't know how many you have. And if it's only 3, you can basically input the 3 FASTQ files, and BRAKER will take care of it quite well. If you, for some reason, get the idea of using all your 20 replicates of your 3 tissues, which makes like 60 alignment runs, I would like align them separately and then merge the BAM file, sort the BAM file, and start BRAKER3 on that. And the main reason is not because BRAKER cannot handle it. The main reason is that these alignment steps on their own take runtime. And I don't know what HPC you are on, but our jobs are killed after 3 days. So if you have a lot of data, it makes sense to like pre-process to the merged BAM file.
Question 2 is, I also have PacBio RNA-seq data, but plan to only use short reads, as BRAKER3 is optimized for them. Given my genome size is around 1.5 gigs, does this seem like a sound approach?
So we do have a special container version for ISOSEQ data. If you look for that, you go to our Breaker GitHub repository in the docs folder. There are the posters from the PAG conferences. And this year in January, I had a poster where I have the Breaker container pulling command for that particular container. You can use ISOSEC data with Breaker. The only change that is in that container compared to all the others is that we changed the string tie assembly command. What we currently do not have is a Breaker version that uses both the ISOSEC and the short read data. Actually, I kind of do have an internal hack on that, but we have never published something. The reason for this is because the string tie assembly is happening in GeneMark ETP. And GeneMark ETP is not my tool. GeneMark ETP is from Georgia Tech, and it has this funny license, right? So you are actually probably legally allowed to alter that string tie command yourself to get both data types in there, but we have not published that. So what you could do if you have both is you once run the long read BRAKER3, once run the short read BRAKER3 and merge them with Zebra. That should actually work decently.
Cool. Thank you very much. Hopefully that answers everything for you, Mohammed. I think the YouTube is on a significant lag as well, so we probably won't know for a good minute or two.
Okay. I see one related question from Stefan in the Discord channel. He says, I ran the long read protocol with ISOSEC data. I got less genes than the species had. How to deal with that? I think what you ran was a deprecated protocol. Like before we released the ISOSEC BRAKER3 container, we also had like in 2023 a protocol for ISOSEC integration. So one of the reasons for your observation is that you used an outdated protocol. The other problem is that ISOSEC sequencing depth is quite often more shallow than short read sequencing depth. And you basically cannot compensate that unless you do deeper ISOSEC sequencing or you have Illumina data. This is why you see fewer gene models. Like how to deal with that, but basically try to run it with short read data if you can get your hands on it. I hope that's helpful, but very expensive probably. So your question was not on that. It was like how to combine it with Zebra. Okay. So Zebra by default, it would take, let's say, 2 or 3 annotation files, GTF files, and 1 evidence file. And if you call it like this, it will do quite a stringent selection on your multi-exon genes. Yeah, it will really only keep the evidence-supported isoforms. If you want to prevent that, and I think that's what your question is really about, you can say, I freeze one gene set that cannot be like depleted of lean models. It has an option, it's called keep GTF or something like that. So you can say, I want to keep this gene set so that I don't lose all other genes, so that my gene set in the end doesn't get so sparse. Sam says, maybe adjust the Zebra config file would increase scoring of single ISO models. Sam, yes, you can spend a lot of time on that. If it would be a straightforward, easy thing, Lars would have published the adjusted config a while ago. Okay, I suggest that we move on to the hands-on thing. Yeah, you're right to laugh tears on that. Now, I hope that you on your own computers see a similar thing to me. And what you need to do first is you click on the B3 on the left in your workspace. Yeah, so click on this B3. It should open up like a file and folder structure. And I want you to click on a file called tutorial MD. That is our hands-on session today. And the very first thing after you manage to do this is that you click Control-Shift-V to get into Markdown view mode. Control-Shift-V. And then it should look pretty. Please give me some kind of indication that you managed to do that, either with an emoji in Zoom or with something on the Discord channel so that I don't lose the audience. Did you manage to do it? Are you there? 1 is there. Okay, good. 2. We have like 25 people hanging on. 3, 4, okay, 5. So we have to wait a little bit so the others actually get there. 7. That's less than 50% of participants. Okay, So I hope I'm not boring you out of your mind. I just think it's actually worth waiting on people for a sec here. Okay. So in case you have not made it here yet, if you have a technical issue, also let us know. You can unmute yourself. Maybe I can quickly help you to fix the problem. I'm also aware that some people will just like sit on the sofa or in the bed because it's nighttime. That's also okay. You're not forced to like really do everything, right? So let's have a quick look around what we have here. You already saw on the left hand that you have this workspace where you can click to folders. What you have now at the top panel is basically our information materials for the session. And at the bottom, you should have a terminal. If you are not seeing the terminal, maybe it's showing you the problems or something, you can click on terminal. We will need this terminal today to execute things, but hold off on executing anything until I tell you to execute something, okay? Good. The slides that I just showed you, they are also included in the folder here. They are also linked. And I want to give you 2 reading recommendations, like for after the session, like if you want to read up on things and this session didn't cover everything. We have a preprint on archive. It's called Navigating Eukaryotic Genome Annotation Pipelines, a route map to BRAKER, GALBA, and Zebra. Tomasz, Lars, and I put a lot of work into making things like easy to understand with a lot of details in there. So you can go back to that resource to read. And another one is that I want to point out that funding for BRAKER has expired, and that's not the only reason why we're not actively developing on that end anymore. Things are going to move towards deep learning. And if you work on mammals, then we have a brand new tool that you can already use, but only for mammals. It's called Tiberius, end-to-end deep learning with an HMM for gene prediction. And even though there's the HMM in the title, this is a deep learning approach. This is a cross-species gene finder that doesn't need the species-specific training step, but it has only been trained for mammals so far. So this is something you can have a look at if you work on mammals. Okay, now we will look at what data we're going to use today. We're going to use a tiny, tiny proportion of the Arabidopsis thaliana genome. All the data we use today have been tailored to make things run fast. Even though fast is still a matter of 8 minutes, what I measured yesterday. What we're not going to run is the repeat masking. It is, however, essential that you mask your genome for repeats. So why is that essential? Depending on the species. Oh, sorry. One second, my kid is making noise here, forgive us. She promised to be more quiet now. Depending on the species that you look at, your genome may consist largely of repeats. I'll give you an example from the diatom project because I just did that the other week. There are species whose genomes consist 80% of repeats. And the problem with these repeats is that some of them look like protein coding genes. So for example, transposable elements, they have things that are protein coding genes, and you don't want to adapt your mathematical model for the gene finder to transposons. Yeah, you want to adapt it to the actual protein coding genes that you're interested in. So repeat masking is really essential. It's also essential for Tiberius. What we typically do is we run repeat modeler 2, but this is quite runtime expensive. I would not recommend you to do that today. When I masked the chunk that we're looking at, I did that on 72 threads to be done in a reasonable time, okay? Repeat modeler 2 will construct a species-specific repeat library. You can combine that with a DFAM library, and then you can run repeat masker to mask the genome. And there was this question in the channel. It was like, should I soft mask or hard mask my genome? We generally recommend that you soft mask. And that means for repeat masker, you have to enable the XS small or X small flag, yeah, in order to get the soft masking. You don't have to use repeat modeler and repeat masker. You could also use red, for example, but you should take care that you get the soft masking. If you have a genome like a vertebrate genome, for example, that is extremely rich in repeats, you can later click here. We have some details on how to fix that for the gene prediction. Yeah, you have to run the tandem repeat finder in an additional iteration. What you generally want to have for these kind of jobs is a lot of threads, and possibly you want to have a lot of RAM. What I typically do to speed this up is I copy everything into the random access memory and execute there. It depends on your cluster configuration whether you're able to do that, but it really speeds up things and it solves the I/O problems that you otherwise have with your storage. Okay, so repeat masking is important. Sometimes I get reports from users who say, oh, but for me the hard masking worked better, or oh, but I hard masked before I mapped my RNA-seq data. This can be the case. What I recommend is like for the general scenario. Yeah, if you are not sure, you can run both and see what's better for your case. Okay. Then BRAKER3 can actually align the RNA-seq data to your genome. It's not a problem. It can internally do that. Today, we're not going to do that, mainly because it takes so much time to run the alignment step. What we use internally is HiSet2, and I would recommend you to use HiSet2, and we also provide the commands here if you want to read up on it. But you could also do star mapping. That would also do a reasonable job. If you use star, please run the 2-pass mapping. So what happens in HiSet2 is that iteratively, while it's running, it adds the intron junctions to like a data structure in order to discover more reads that map to those intron junctions. Star is not doing that. So for star, you run it once, then you have the database of the intron junctions, and then you run it again with that database. Yeah? So that's why 2-pass mapping makes a lot of sense. If you prefer star, they're both really good aligners. Internally, we'll always use the HiSet though. Today, we're going to use a precompiled BEM file where we have run the mapping in the past to save us a bit of time. It's sitting in the container, we have access to it. Now, this here covers like a lot of the stuff that I told you about in the talk. Let's have a look here at the cell where I show you an actual BRAKER3 command. So what we have in here is how many threads you're going to use. I put that in a variable because my bet was that some of you maybe booted the Git pod with 4 threads only because you booted before I said you should use 8, right? So if you use 4 during booting, you should decrease that number. Yeah, you can like double click on it here, and then you can just type in there and make it a 4. It's not a problem. You can go back with the Control-Shift-V into your Markdown view. Then what happens in here is, in case you later want to repeat what we did today, is that we first remove the output folder because BRAKER3 will otherwise have trouble if you have stuff in the output folder. So if it's there, we remove it. And then we fix what's our protein input. In a real life case scenario, if I want to annotate Arabidopsis, I would download the Viridiplante OrthoDB partition. However, if we use this big OrthoDB file with our small genome data set, the runtime will be 30 minutes. So your job will probably have finished right about when we finish the session. That's why I'm very grateful that we got a smaller data set compiled. Trying to remember who did that. One of the really amazing people from the Chesky Krumlov workshop did it. I'm so sorry that I cannot give proper credit right now. This is not our data set. Anyway, this is like a custom artificial small fake data set that we're using today. In real life, please use a big data set. And as I said before, you do not only have to use the OrthoDB partition, but if you have reliable protein sequences of the 2 sister species of your target organism, download them, make the FASTA headers simple. We have a script for that. It's called simplifyfastaheaders.pl. And then you just concatenate with the cat command to the OrthoDB partition to increase the accuracy of Breaker a little more. You can find all the OrthoDB partitions on our website. OrthoDB is originally maintained and provided by Yevgeny's group, the OrthoDB team, but we asked him whether we can host the partition dataset on our website since they stopped hosting partition datasets with the ODB 11 version. If you cannot find your partition in there, drop me an email. I can tell you how many genes are or proteins are available for your partition. I think we created all the big partitions. Usually it's a problem to find data for the smaller ones, but I can look into it. So these are our inputs. We have the precompiled BEM file. This is from Illumina short-read data. We have the artificial protein data set. It's defined here. And if we want to call Breaker, we always call the Breaker.pl script. It doesn't matter whether you use BRAKER1 or BRAKER2 or BRAKER3, it's always the same script. You can, but you don't have to provide a working directory. If you do not provide a working directory, it will make a directory called Breaker. Now I call it BRAKER3 just to show you you can make your custom directory name. We here have a soft masked genome chunk from Arabidopsis. It's sitting in our container. This is our BAM file specification. This here is our protein sequence database specification. It's a FASTA file with proteins. And then this here, you don't necessarily have to provide. I can also delete this, the job is still gonna run. But I want to point out to you, depending on how you install Breaker, and if for some reason you do not use our container, Breaker will need the path to the Augustus and E-training binary. Yeah, that's what is in the Augustus bin path. And it will also need the path to the Augustus scripts. That's in the Augustus scripts path. In the container that we use today, this is all in there. So this could go away, job would still run, but depending on how you install yourself, it might make sense to provide it. And then we provide the number of threads we want to use. Not all steps of Breaker take full advantage of the threads, which is kind of sad, but the really expensive ones, they do. And then optionally, you can provide the Busco lineage. I have heard reports from some people from countries where the Busco server is behind the national firewall. This is kind of a bummer. I think you can like pre-download the Busco lineage files if that is the case for you. You find some GitHub issues on that. People have managed to work around it. We usually assume that you're not sitting behind the Great National Firewall, and then this should just work fine. It will automatically download the lineage file for you from the internet. So this requires usually an internet connection. And then here are two options that you do not want to have in your real job. They're just here to decrease the runtime today, okay? So we have here switched the GeneMark maximum intergenic region to 10,000, which is a ridiculous choice. It just makes it fast now. And we have also here set a step called skip optimize. This cuts the runtime by probably 6-7 hours. You do not want to skip Augustus optimization for your real genome. The difference in accuracy is usually ballpark numbers 1 to 3% on gene level. And you can say, okay, those 1 to 3% don't matter to me. That's okay, fine by me. But if you want to have the highest accuracy, you want to delete both these options from your real job. Now, we're going to move into our, we are already in the workspace. That's good. We're going to copy all the stuff here with your adjusted threads, and we're going to put them into the terminal window, and we're going to hit enter. I'm checking whether there were a lot of questions or no, these were all thumbs up. No, there are questions. Okay. While you let this run, yeah, job is running now. It's going to run for a while. I will look at the chat. There's a question for repeat masking. How about Earl Grey for speed? Did you recommend this tool? It's actually the first time I hear about Earl Grey, so I have no idea whether I recommend that tool. Can't tell you. If you say it's good, I'm going to check it out. Thank you for the recommendation. The fast thing that I was aware of was the red mask. Okay, then how to do masking good? Is red this interesting post? Is that a good approach or not? I think you are referring to red mask, but I will quickly click on the link to see what it actually is because, okay, I cannot get out of the chat to do that. Let's see, what is that? Okay, this looks like a pretty long thing. I cannot tell you without reading it whether I recommend that. I'm sorry. Red is red mask. Red mask has repeat libraries for a very small number of species. However, people at the EBI keep telling me that it performs quite well across a wide range of species, but not always. We have seen examples where red mask would severely undermask. That does happen, and then we have to do repeat modeler. I still do repeat modeler out of routine. There was a follow-up on that.
For Earl Grey, so Earl Grey is a transposable element annotation tool, and I'm pretty sure they still use repeat masker and repeat modeler for masking.
Good to know. So there are a number of tools for the repeat annotation. So there is a difference between finding them and saying what they are. For the gene prediction, we don't care what they are. We only care that they were found and masked. So these are probably diverging questions. Okay, thank you for putting that into context. There was a question in the chat about the GeneMark max intergenic parameter. What would you use? Like for a real run, you would not specify it. It is set to a reasonable default value. You would simply not specify it. Then there's another one. Can you comment on the accuracy species-specific training models of Augustus selectable as a parameter versus the custom run specific training model generated during the breaker run? I've assumed the custom training model is more accurate. Nice, Sam. So Augustus is released with a number of species-specific parameters. The ones that have been there for a very, very long time, like since 2008, 2009, they were basically manually trained by people. Some of them were done by Mario, some of them by external collaboration partners. There are some parameter sets that are of amazing quality. For example, the human parameter set is really, really good. A breaker run will not do better on human. Also, the Chlamydomonas reinhardtii parameter set, it should be really amazing. But this, it should be really amazing, does not hold for all of these Augustus release sets, because some of them were actually generated with Breaker, particularly the ones that I added in recent years. They were Breaker2 generated mostly. So it kind of depends, right? So if you do human, use the Augustus parameters. You can even use them for other mammals. They work really well in mammals generally. If you do fruit fly, you probably also want to use them, but it's not a general thing. Like some of them are breaker parameters. So maybe you have to run both if you really want to know for your particular species. Okay, then there was another one. Have you reviewed the 10% or 20% of the genes that Breaker or Galba failed to predict accurately? Are these any common themes or issues with these genes? We have not spent much time in classifying them. There are some issues that can be spotted, but no, we have not looked much into details there. Then there was, early on, I was using the Docker container a lot, but later I used Singularity, which saves me a lot of time with the key expiring every 3 months. I understand that's okay. Okay, Stefan. There was a switch in how to handle the licensing of GeneMark, and that switch happened like many months ago. So in the past, the GeneMark team always required us to ask users to download a license key from Georgia Tech website. That was before they went to the other license. This now changed both for the Docker and the Singularity container. Our Singularity container is basically the Docker container. We frequently update it, and if you encounter problems, I encourage you to rebuild your container. There's a bit of a tricky thing if you are not an experienced Singularity user. So if you originally built the latest image, you may have to specify a version tag, because if it already has latest, it will not know that latest is now something else. But we put effort into tagging properly. So now it's like v3.0. I don't know, something. So you can rebuild those containers, and Docker and Singularity are the same, and the license key is not required anymore. Okay. Are there any specific parameters in BRAKER3 for gene-dense genomes where fused RNA-seq evidence is common? Nope, we don't have specific parameters for that. Do you have a link to the correct protein database? Good to know. Thank you very much. The protein database link is in here. Yeah, I hope you can see it in the screen sharing. You can click that in your web browser, and then you find the OrthoDB partitions that we have built. Okay, that was a comment on the Earl Grey. Someone said, I do this, I use Earl Grey. I think Earl Grey does a better job to find LTRs. In the latest version, it can give a soft mask genome. I can give a soft mask genome, which I then use directly in Breaker. Okay, so Earl Grey maybe does a better job on LTRs. By the way, a repeat modeler sometimes dies on me on the LTR step, so thank you for letting me know. Like, it exceeds our runtime limit frequently. Okay, Chiara actually put the link into the chat. And then there was one more. To use protein databases for species that are more or less closely related in the absence of reference proteins. Is it better to use the RNA-seq libraries directly, or to assemble them, pass them through transdecoder and obtain proteins. No, no, no. Don't assemble them and pass them through transdecoder. So we use this protein database to denoise the ORFs that we call in transcriptome data. If you use transdecoder in assembled transcripts, the output is going to be very noisy. You're basically putting the denoising step a bit to the absurd space if you do that. I know this for a fact. My bachelor student is working a lot with transdecoder for this right now. It introduces noise if you do that. I'm not sure what scenario you have in mind where you cannot find a protein database. I can give you some advice that sometimes it helps to combine things. For example, when I did the diatoms, I did not only use straminophiles, but I put in the Viridi plantae, even though I know diatoms are not higher plants, but it helps, it provides sufficient material. Okay. I get a bit confused by the ORTHODB 10 or 11 because Busco and Olmark are still using ODB 10. How can you compare the output of BRAKER? These are two different questions. One thing is, I don't know when they're going to update the Buscos. It's probably going to happen. I don't know when. I think Olmark is not built on the ODB. OrthoDB. I think Olmark is built on, is it ensemble proteins? I would have to reread the paper, but it's not OrthoDB dependent, I'm pretty sure. So those marker gene sets, they are on their own pretty independent from BRAKER. The one thing where it can get confusing is if you use the Buscos in BRAKER. Like if you want, for example, to compare how well does Van Annotate discover Buscos and how well does BRAKER discover Buscos, then if you just supply the Busco lineage option to BRAKER, it would be like an unfair advantage to BRAKER compared to Van Annotate. However, Van Annotate uses the Buscos in the training assembly in the first step, but that's another story. So if you want to benchmark maybe different pipelines, yeah, and if you really care how good is, for example, Augustus at detecting Buscos, then you should not provide the option. But if you are a user who's interested in having a complete gene set, then there is no reason to exclude genes that Augustus predicted and that we keep in the final gene set because they are similar to Buscos. Like you want to have them, you don't want to drop them, and we don't have other evidence for them. That's why we do it. I'm not sure whether that made it less confusing for you. I hope it did. Okay, as you can see, my job is still running. And I don't see any other questions at the moment.
I have one in YouTube.
Yeah.
Mohammed Salman is back. Would you use the no low flag and no RNA option in repeat masker?
No, I do not use them. No. Maybe we should, but we don't. Okay. I saw this coming, that this would take a long time. And this is why I actually pre-computed all the results for you. And while we keep the job running here, yeah, it's how long are we? We started 9:12. It should be done actually in a minute or two. We can have a look at a folder with the BRAKER3 pre-computed results. Here, this one. I hope you saw that popping up on the left. This is the typical most important content that any BRAKER output folder would have. And the first thing I want to draw your attention to is the log file. Yeah, all these folders, they should have a BRAKER log file. And one thing you want to make sure of is that your job finished okay. So you can open that. This is how it would look like. And scroll to the very bottom. Whoops, here. And it should say BRAKER run finished. If it doesn't say BRAKER run finished, then something died on you. You may want to look at where it died. Okay. So that's the BRAKER log file. Trying to close that. Cannot close it. Everything is stuck here. I hope things don't get stuck for you. At least I can switch back to a different tab. Nice. Okay. Then the next important thing I want to draw your attention to. So the breaker log, if something died, it will have some kind of a message. But the error output files of individual tools, they end up in the errors folder. Yeah, the last step that was executed will probably have piped the error message into a file that's in the errors folder. And if you want to see what actually happened, you need to look at that file content. And also if you put an issue on GitHub, like, hey, it died at that step, please do the little effort without us have to ask back to check what's in the output files, because I don't have a glass ball. I don't see the content here. That would be really cool if you looked at it. Oh, by the way, now with 12 minutes, yesterday it was only 9, my job finished. As you see, I get a warning message. It says number of reliable training genes is low, 114. Recommended are at least 600 training genes. The 600, we discussed that before, is a bit of an arbitrary choice. Yeah, but this is why we set Breaker to actually issue a warning. If you have more, it will be better. So now we have two output folders. We have the BRAKER3 pre-computed files and we have the BRAKER3 files, but we will continue with the pre-computed ones because that's what I prepared the code cells for. And then your gene models. The main output of Breaker is in the Breaker GTF file. If you run it, you can run it with the Breaker with minus GFF3 flag. You will also have a GFF3 file of the same gene models. However, I usually output GTF and then I convert later. It's kind of a preference of myself. I like to convert with other tools. And then you have a coding sequence file. That's a FASTA file with the predicted coding sequences, and a protein file. The .aa file is basically your protein sequences in FASTA format. If your FASTA headers were long and complex because, for example, you directly downloaded your genome assembly from NCBI datasets, they have a lot of stuff in the headers, then Breaker will have shortened your headers in the FASTA file because some tools cannot digest the long headers with the white spaces. Then you have a map file here that says this was like what you called it, this is what we call it now, sorry. Okay, the apology is only oral, it doesn't say sorry. And then you have the species folder that will contain a copy of your Augustus parameters that you optimized in this job. You have a GeneMark ETP folder that contains all the GeneMark ETP output. And you have an Augustus folder that contains the Augustus hints gene models. Now, what I want to tell you is how you can inspect these results. Give me 1 second. My kid wants something. Okay, we have a life-threatening situation. I promise I'll be back. Okay, so, sorry, someone literally died. It was too late. A hedgehog fell into our pool. I'm very sorry for the hedgehog and that I had to interrupt this. Poor thing. But it's very cold already. Good. We were talking about what to do with output files of Breaker. Now, if you copy this code cell here, you will move into the folder B3 pre-computed results, and what it will do, it will show you with ls minus lh the size of the different output files that are most important for a Breaker run. I showed you already, one important output is the Breaker GTF file. That's this one here. You see in this case it's rather small, it's 393 kilobyte. If ever you're not happy with the Breaker gene models, like in your genome browser, you see, I don't know, I'm missing a lot of gene models, or you see it in your Busco output, you can go to the files that were used to build that file. And that would be the GeneMark GTF file here. It's like 401 kilobyte here. Or and the Augustus Hints output file. That would be this one. This is why we look at them. These are the files that were used to build the final output. And sometimes BRAKER3 will not deliver the optimal result, but you will find the genes you want to find in the other two files. Yeah, that's why we look at them. Okay, now we will move a little down here. We will look at what files do we need to, for example, run OMARK or run BUSCO, because you want to run those benchmarks on your gene sets. If you want to run BUSCO, you need the protein sequence files. The protein sequences are in the BRAKER AA file. They are in the Augustus Hints AA file, and they are in the GeneMark AA file. And before you think about doing anything to combine these GTF files in a different way, the first thing that you do is that you look at these protein files, like, is there any hope? Yeah? If BRAKER, for example, dropped too many gene models and there are no more protein sequences in the input files, then you cannot restore anything from that. But in most cases, you will see, and also in this case, that Augustus had obviously a few more protein sequences. No, it didn't. In this case, it didn't. It did not have more. But sometimes you see that GeneMark or Augustus had a few more sequences than ended up in the final gene set. And if your final gene set is too small and you see that there are more protein sequences in these two other files, then this is an indication that possibly you want to either use the other files as final output or maybe rerun Zebra manually to obtain a different gene set. Now, the sad thing is that the GeneMark protein FASTA file, this one here, it doesn't natively show up, which is why I give you the command here to use the get anno FASTA from join genes script, where we basically use the GeneMark GTF file as input with the genome file to generate that file. Yeah, so BRAKER output file is automatically there, the protein file. For Augustus, it's automatically there. For GeneMark, you always have to generate it, but it is rather easy, as you just saw. I'm still trying to collect myself after the death of the head shock. I'm sorry. Okay. Now, I told you before, you can compute descriptive statistics, and there are many ways to do that. And I actually encourage you, someone please write a good pipeline to do this. We have a very simple script for that at the moment only. We will obtain that script now from the internet. It's stored in the Galba repository. We call it Analyze Exons. We make it executable, and then we will apply it to the different GTF files to get a description. So the output was done incredibly fast here. What this script gives you is a number of transcripts. And this is a bit of a confusing phrasing. I made the script in order to assess the mono to multi-exon ratio. If I have many alternative splicing isoforms, I do not want to use all of them to compute that ratio. I want to use one isoform. Yeah, so this number of transcripts here, it's actually a number of genes. I take like the longest isoform here. I don't use all the isoforms. And then it will give you like the number of exons in all transcripts, like what is the largest. Here we have a large gene with 30 exons. It gives you the number of mono and multi-exonic ones, and it gives you this ratio. As I said before, people believe it should be around 0.2, so it's a bit too high here. And it also gives you the values you would typically use for a box plot. And you can look at this like for all the different gene sets. What we see often is that for Augustus, this ratio may actually be increased much more than you see it here. Here it looks almost identical, but often it's a lot higher. So Zebra is doing for us like a denoising step also to quench that a little bit. Okay. So this is one way how you can quickly access results. Now, if you have the protein files, you could also run BUSCO. That's straightforward. For OMARK, I encourage you to go to the GitHub repository and look at how they want the alternative splicing files to be nested. Like you can make a text file that lists basically the names of the proteins that belong together so that OMARK can handle them correctly. Okay. Now, I said before, one of the most important things is that you visualize your data in a genome browser. You can do that with any genome browser, of course. Please, Hannah. we have another hedgehog dead. Is der auch tot? So we have 2 dead hedgehogs this morning. Okay, we'll have a funeral later today. I'm so sorry. Genome browser. So one of the things you can do easily with a Breaker output is to build an assembly hub. An assembly hub is a local data structure that you place on your web server, and then you can access one of the key configuration files of that from the internet, from anywhere, and you give that link to the UCC genome browser. Yeah, so this is something where you don't have to install a lot of software. The browser rendering is basically handled either in University of Santa Cruz or it's handled in Bielefeld, where they have the European mirror. Yeah, so we have a script to automatically from any Breaker output make this data structure, and it does like everything, like handling all the different evidence tracks and so on. This is one reason why I actually do recommend using this, but you can also use IGB or something. So I will quickly show you that this actually works. It will always download 2 AS files from UCSC in the beginning, and then it will simply compile the data structure. Now it even tells you what to do next. Now there is a folder, it's called AT chunk, because we have only the Arabidopsis chunk here, and it tells you that you could copy that to your web browser and then check what is the correct link for your hub.txt file and put that into the UCC genome browser to look at your data. If you want to do that, we have this particular hub already sitting on our web server, and you can try that later if you like. Here I'm just showing you a screenshot. So the whole reason for doing this again is because you want to compare our gene predictions. Here they are blue. This is one BRAKER3 gene with the different evidence tracks. And what we typically have if we run BRAKER3 is protein evidence. So these here are chained CDS that come from protein to genome alignments. And we want to check whether this also matches with the intron coverage. That's the cyan thing here. And we may also have evidence, in this case we do, for start codons. We may have it for stop codons. Here we don't. So you want to look at this for a bunch of loci. And one of the questions people always have is like, how do I know where to go? So what I typically do is I look at the long sequences, and you can extract the information on what sequences are actually long with this command here. It's a simple combination of script from Augustus, summarize, ACGT content, and grep, and head and sort. And this gives me now a number of faster header names. Like typically here we would look at X1, for example, and at X5, and jump around a little bit in that chromosome or in that context to look at gene models. Now, coming to the end of the walkthrough, if you want to run Breaker locally, you can use our container. Yeah, if you have never pulled it before, pull the latest. If you have pulled it before, like go to our Docker Hub page, look at the tags, like which version are we at the moment, and replace the latest with a specific version so your computer knows that it has to update it. So this here is a command you can use with Docker to basically see the same environment that we saw here. You will not end up in a VS Code. I think you end up in a Jupyter instance there, but it's going to work the same way. Most people don't have root permissions. They cannot do Docker. So you can also use Singularity if you have it to build our container. And you can call Breaker with Singularity. If you have never done that before, there are a few important things. So the Singularity image is a frozen thing. It's static. You cannot edit anything inside of it once it has been compiled. So if you want to write something, you have to write it to a writable location. And for this, you have to tell Singularity where it can actually see things. This bind command, it's essential. You have to basically mount the places you want to be able to see to your container, both for reading and for writing. Yeah, and you can like bind more than one directory, no problem. You can have several bind commands, but if you want to see data that's not in your container, or if you want to write places, you have to use the binding command. One thing that some users have said is that if they run the Singularity container in their standard environment, they get into a mess with their BashRC contents. Yeah, they have exported things in their path and things got overwritten and so on. So you can inhibit that. Yeah, we have our own path environment in the container, and you can basically stop using your external definitions with the clean env command. I usually don't have to run that, but some people have to. So this is how you can run Breaker from Singularity. Okay, few troubleshooting things. BRAKER3 died. What can you do? First, maybe check why it died. Most of my jobs die because our HPC has a problem, and then it's not a breaker problem. For example, a switch burns or something. Then just restart it, delete the output, restart. If it really died, which happens regrettably, check where it died. Like there will be a step in the log file, and there will be output and standard output files that have possibly some contents. And you can either try to figure it out on your own. You can search our GitHub issue list for keywords, or you can open a new issue. We're trying to address issues on GitHub, but we are like a small team that's now putting a lot of effort into the Tiberius project. So we're a bit slow on responding. Also, we cannot fix GeneMark issues. What I hear a lot is like, oh, I got too many gene models. I got 80,000 or 100,000. Oh, what to do? The thing is, first of all, you have to figure out how many gene models you actually expect. If you have a polyploid genome assembly, maybe for 80,000 is not wrong. If you have like 2 copies, could be a realistic thing. If that is not the case and you're really sure something is wrong, it usually doesn't make much sense to tweak with breaker parameters. You can see whether there's a different pipeline that does a more decent job for your genome. Yeah, sometimes we just cannot solve it well. I will not have golden advice for you on how to address that. There are other pipelines like EASL. There is a session by Jill Vagerson on that, I think. There is Maker. Maker I often do not recommend, but sometimes it does a decent job. Yeah, Fun Annotate also. Generally, we think that our pipelines work better, but sometimes it may do just the job for you if you got a problem with way too high gene count. One thing you can log into is repeat masking if your gene count is too high. What you can do, for example, is you do reciprocal blast with the breaker output proteins. And if you then sort your hits and you see that, I don't know, there are like 30 proteins that basically all hit each other, you can also blast them again to see what it is. What I see quite often is that they are retroviral proteins or something. And if this is inflating your gene count, you can actually improve your masking. So in that case, you can quench it by just doing a better job on the masking. And we, even in this notebook, give you some advice on how to do that. Okay, then there's also the underprediction problem. Some people complain that they don't get enough gene models. In BRAKER3, this was a common complaint because BRAKER3 really filters in the zebra step. It really discards a lot of stuff that has no evidence. As I said today, there are the intermediate files. You have the Augustus file, you have the GeneMark file. Maybe one of them is the better file without the filtering for you in your specific case where you have so little evidence. You can also run Zebra to recombine, to like freeze one of them and just add some gene models on top of that. But I think we addressed this mostly with the integration of the Busco lineage. So this kind of made this problem a little bit go away. Okay, then how many genes to expect? Well, look at the relatives. It can be very different. There are protists that have less than 10,000 genes. They're fine. And there are polyploid genomes that have a lot of genes, more than 100,000 could be seen. So there is no answer for all. The isosec question we discussed, we have a possible solution for that. And then, yeah, why do we not reply? It's basically because your problem is hard to solve. If it's easy to solve, you usually get a reply super fast. But if we're like not really sure what's going on, then it's gonna take a long time. Community is great. Some people are answering questions for others, and I'm super grateful to them. And what to do? If you found no answer, please go to GitHub. Don't start sending individual messages, please. I really like being in touch with people, but if you put it on GitHub and not to my personal inbox, there's a higher chance that people see your problem. Okay. Now let's go back to the questions. The paths for the bind are the same. Okay, I go back to that and I elaborate a little bit. What you put in the binding command is the real path to a folder on your system. It's not going to be directory you may want to mount. It's going to be, for me, for example, home Katharina Breaker genomes. Yeah, that's going to be my folder where I keep my genomes. And then I type this here, and you can either give it a different name here, or you give it the same name. If you give it a different name, like this is like, where is it coming from? This is where is it mounting to? If you give it a different name, you also have to give it the correct name in your breaker call. I like to put these two things in the same way so that I don't confuse myself and don't get a headache. But you can like call them differently. And yes, if you have multiple things you want to bind, you just like put a space here before the breaker and you make another bind command. You don't also have to type the -- bind. You can use -B. It's going to do the same thing. Now I'm trying to find there are the questions on the Discord channel. So, since for most distantly related species, it is almost impossible to map directly, my idea is to sample the RNA-seq data and use them as proteins. As I said before, I don't recommend doing that. I also work on genomes where there is not so much data in the databases. I work on protists and so on, and I would not recommend to do that because the transdecoder called proteins are hell of noisy. Transdecoder will do things like calling multiple ORFs even in one transcript, and we know that operon expression is usually not a thing in our eukaryotes. That's one of the examples where it goes wrong. Okay, Stefan had a better runtime than I did. Thank you for reporting that. Sam says, Augustus Hinz GTF versus Augustus ab initio GTF. I forget, does Augustus Hinz GTF include the ab initio? No, it does not. Something we did not cover today. Breaker has a command line flag. It's called -- Augustus _ab _initio. This is a flag that we mainly introduce for internal evaluation purposes, because we as developers are interested in the accuracy from the step of ab initio and Hinz integration. I think for most users, this flag should not be super interesting, because we know for a fact that the ab initio gene models are going to be much worse than the Hinz supported gene models. Augustus with Hinz will contain gene models that have Hinz support, and it will contain gene models that have no Hinz support. So from a user's perspective, I think there should be no reason to invoke the ab initio flag unless you are like into evaluating the intermediate steps of the pipeline. Okay. Yeah, I'm sorry about the hedgehogs too. And my daughter said she even saw a third hedgehog. Is it dead too?
Was?
Is der auch tot?
Nein.
Okay, the third hedgehog is alive. I swear to God, we usually don't see hedgehogs. I don't know what's wrong today. At least one is alive. So another question. I'm getting less stop-start codons than genes. How can that be possible with combining isosec and short-read data? Augustus predicts genes that will expand over your boundary of the sequence, or that will go into N regions. Like if you do scaffolding, sometimes you have two different contexts that will be connected by some spacing region consisting of Ns. And Augustus can predict partial gene models. Yeah, it can predict things where one end is missing. So it can happen that you have like a larger number of gene models than you have of start codons and stop codons. You can suppress that. If you don't like it, you can suppress that. There is a breaker option called -- Augustus args, I think. And there you can say you want to predict only complete genes with Augustus. GeneMark doesn't do this thing. It's only Augustus that does it, so you can suppress that. Okay. Good. These are the questions that I can see. Do you have some on YouTube?
No, nothing. And I think this was such a comprehensive session that you've answered all my questions already.
I'm glad to hear that. My daughter is commenting, it's really weird with the 3 hedgehogs today. She's holding up
It is
sign.
pretty weird, yeah.
Okay. Yeah, I feel kind of embarrassed that will go on YouTube, but whatever, maybe it will make someone feel for the hedgehogs.
We can always cut it out in the actual one. So You don't have to worry about it forever as the hedgehog woman.
Okay. Good. I see someone is maybe gonna post one more comment. Okay, these are like, thank you for the sessions. Well, thank you for being a great audience today. Feel free to reach out. Some discussions can probably be taken to GitHub, so maybe Lars can also jump in on commenting things. Yeah, and we all feel for the hedgehogs, and I will, after the session, dive into a little funeral service in our garden. I hope I find a shovel. Okay, people are fading out, so I will also maybe keep an eye for a few minutes on the Discord channel. If questions show up there, I will reply in a written manner. I will now say goodbye and stop the screen sharing, and also wish everyone a good day. And thank you, Damon, for organizing this. It must be really stressful to organize all these sessions.
Just a bit, yeah. No, thank you very much for offering your time up to do such a session. It's been fantastic.
Okay, then I wish you best of luck with the next one. Bye-bye.